we will spend the next few minutes reviewing some very basic concepts related to population genetics. And so we're going to focus primarily on Hardy-Weinberg and also briefly review some of the concepts related to evolving populations. Please keep in mind that this will be very brief, and I encourage you to instead spend most of your time reviewing problems related to some of these concepts, as this will be the focus of what I would expect you to demonstrate an understanding of on our next exam. So if we think about population genetics, we can think about tabulating and making predictions on the frequencies of various things, for example, genotypes and alleles. So for example, if we have a hypothetical population of frogs like we see here, and let's say we have a total of 16 frogs in this population, and if we know the genotypes of each of those frogs, we can tabulate those, and it's easy enough, of course, to then figure out the number of various alleles. So for example, in this case, we're talking about a gene that's illustrated by a big A and a little a. Think of them as dominant recessive. That's perfectly fine. And if we have the following genotypes listed here, so 5 of them are homozygous dominant, 8 are heterozygotes, 3 are homozygous recessive, then it's easy to then figure out the number of alleles in that population, right? So we would say, well, there are 18 big A alleles and 14 little a alleles. And of course, it would be easy enough to then convert that to a frequency of each of those 2 alleles. So information on genotype frequency and allele frequency can be used to make some predictions. And that's what this chapter is all about, understanding the relationship between the frequency of genotypes and alleles and seeing the extent to which the frequency of those of alleles change in populations generation after generation. So for example, if we think about evolution at the most basic sense in terms of the change in allele frequency over time, we can think about a population that is not evolving. In other words, alleles remain constant generation after generation after generation. And this was sort of formalized, if you will, by 2 scientists, Hardy and Weinberg, who basically figured out that if you have a population that is not evolving, in other words, allele frequencies remain the same generation after generation, then certain conditions would have to be met. Namely, you can't have any natural selection taking place. You can't have any gene flow, so no movement of individuals into the population or out of the population. You can't have any mutations occurring. Mating has to be entirely random. There can't be any choice in terms of what organisms mate with other organisms. And the 5th one we'll mention here, a large population size is needed because, as we'll see, genetic drift, which is a random change in allele frequency, is especially prevalent in small populations. So obviously these conditions, or at least all these, are never met in nature. So it's very, very difficult, if not impossible, to think of a population that does not violate at least one, if not more, of these Hardy-Weinberg assumptions. Okay, but the whole point of this is using this as a way to measure the extent at which allele frequencies change over time. So Hardy and Weinberg came up with some basic equations that we'll point out here in just a second. But before we do that, I'm not going to spend a lot of time on this figure, but this is a nice figure that illustrates sort of the rationale behind Hardy-Weinberg equilibrium. So I encourage you to pause the video if you want to, just to sort of work your way through this and see the logic behind this. But in a nutshell, it goes something like this. So imagine a gene that has 2 versions, maybe a dominant and recessive, for example, but simply A1 and A2, or in this case, a red and a yellow. And let's say initially if we have a population where the frequency of the red is 70% or 0.7, and the frequency of the yellow is 30% or 0.3. You can then imagine those individuals in a population mating and having offspring. So you can see the chances of forming an offspring that has both of the red alleles would be 0.7 times 0.7 gives you 0.49 or 49%. The chances of having an offspring with both yellow alleles would be 0.3 times 0.3 or 9%. Or having offspring that are red-yellow or yellow-red would essentially be 2 times 0.7 times 0.3 or 42%. Then we have this pool of offspring which would then mature, grow up, and they then could have their own offspring. And then if you run through the numbers again, essentially what you find is that in the gene pool of the offspring, essentially the frequency of the red allele remains at 0.7 and the frequency of the yellow allele remains at 0.3. So the bottom line is that the frequency of the alleles remains unchanged generation after generation. Again, that's if all of the assumptions for Hardy-Weinberg were met. Okay, so let's use some of these concepts illustrated here to quickly review the Hardy-Weinberg equations. So essentially, it doesn't matter which letters you use, but they tend to use P's and Q's. So the frequency of P plus the frequency of Q has to equal 100% of your total population. So we'd say P plus Q equals 100%, or simply 1. And that just tells us the frequency of the two alleles, P and Q, big A, little a, however you want to think about it. If you want to think about the frequency of genotypes, then we can think about P squared plus 2PQ plus Q squared. And again, think back to the previous screen for the logic behind this. So P squared, you might say, well, it's the frequency of homozygous dominants. Q squared, the frequency of homozygous recessives. And 2PQ, that's the frequency of heterozygotes in your population. Again, they have to combined equal to a total of 100% or simply 1. So those are the basic equations that we can use for calculating predictions of alleles and of genotypes in populations. Obviously, as we said, Hardy-Weinberg assumptions are rarely met in nature. So I'm not going to dwell on details here because you hopefully have seen these before. But again, think about natural selection. Of course, we're talking about anytime there's selection for specific phenotypes or genotypes in a population. So this, of course, was sort of formalized by Darwin and elaborated by others and so forth. But basically, we can say that natural selection has the following components. So Darwin basically said that in order for selection to occur, there has to be more individuals produced in each generation than can survive. In other words, there's resources are limited, and you might have competition for resources, and there might be disease and so forth. Okay, but again, more individuals are produced than can survive. There's some variation, some phenotypic variation among individuals in some trait. Also, that variation has to have a genetic basis so that it can be passed on to subsequent generations, and there might be fitness differences among individuals with different phenotypes or genotypes. So again, those are the basic components of natural selection. Think about all the assumptions for Hardy-Weinberg that we mentioned previously. And again, we can just quickly point out that, again, they rarely are met in natural populations. So gene flow, for example, moving alleles from one population to another. Very common, occurs in both animals and plant populations. So it's a very common natural occurrence of sexually producing populations. Genetic drift. Genetic drift refers to a random change in allele frequencies over time, not necessarily due to any selective advantage. So for example, we can think about the founder effect and genetic bottleneck. These are both examples of genetic drift. So founder effect, imagine you have some initial population illustrated here with a certain frequency of, let's say, 3 different genotypes. And then just by chance, if you have a small number of individuals that leave the population and establish a population elsewhere, again, just by chance, those individuals might have different frequency of alleles than did the original population. So they can then go ahead and create offspring, which ultimately, again, might have different frequencies of alleles than the original population. Not because there's any selective advantage to the theoretical red and purple alleles here, or disadvantage to the blue alleles, but again, it was just a random chance event. Likewise, genetic bottleneck, similar idea, but the basic idea here is if a population is decimated by some catastrophic event, again, just by chance, some genotypes might survive more highly than others, again, just by chance. So again, these both represent examples of genetic drift, right? Random changes in allele frequencies over time. In addition, obviously, mating is rarely random, organisms are always, of course, making choices about who they mate with. So, for example, this phenomenon referred to as assortative mating. So here we see blister beetles demonstrating this concept. The idea is that you tend to see with assortative mating individuals mating with other individuals who have very similar phenotypes. So that's what we see here, big beetles mating with other big beetles, small ones mating with other small ones. Also, this concept of inbreeding. To the extreme extent, you can imagine inbreeding as occurring when individuals that are hermaphrodites, they produce both sperm and eggs, basically self-fertilizing. Okay, the less extreme example would, of course, be individuals mating with close related individuals. But let's think about the extreme case where we have individuals self-fertilizing. So if you imagine that scenario, you can ask the question, well, then what happens to the frequency of homozygotes or of heterozygotes as a result of inbreeding? And what you see is illustrated here. So if you're an individual that's homozygous for a particular gene, perhaps homozygous dominant in red, maybe homozygous recessive in blue. If you're homozygous for a particular gene, again, if you self-fertilize, all of your offspring would be homozygous, right? What if you're an individual who is heterozygous for a particular gene? Well, what you can see is, yes, on average, you're going to produce some homozygote offspring, some heterozygote offspring, but you're more likely to produce more heterozygotes over time than you would homozygotes. And so you can imagine this situation generation after generation after generation. That would result in an increase in homozygosity, right? So homozygous, dominant, and recessive, they increase in numbers while the frequency of heterozygotes decrease in numbers. Okay? And the impacts of that can be seen as a result in inbreeding depression. They're very common in many plants and certainly common in many animals and so forth. This is just a representation of inbreeding depression. So Florida panthers, these are an endangered species. And what this illustrates is if you mate Florida panthers with other Florida panthers who are mating closely related individuals, their offspring just have a hard time surviving, compared to the situation where if you mate Florida panthers with panthers from Texas. So you're just increasing genetic diversity. As a result, those individuals are simply more likely to survive, right? So basically the difference between the purple line, where you're outcrossing, and the orange line, where you're crossing close related individuals, that difference essentially represents inbreeding depression. Recall that another component or assumption of Hardy-Weinberg equilibrium is no mutations. Obviously, mutations do occur, right? They're part of nature, and mutations essentially represent the source of new alleles or of new genetic information within a population. Mutations are rare. And it's rare that they might end up in gametes and so forth, but obviously they do occur. So on their own, they might have a very small effect on allele frequency, but mutations combined with all of the other processes we just described can have a potentially big impact on the evolution of populations. So that concludes our very brief overview of population genetics.